we realized that there are many, many studies that showed that a high uric acid independently predict hypertension, and it independently predict obesity and diabetes and all these things. And so it looked like it was really important.
Hello, everyone. I'm Dr. David Perlmutter. Welcome again to The Empowering Neurologist. On this program, we focus on metabolic issues, things like diabetes, insulin resistance, dyslipidemia, obesity, overweight, hypertension. All of these things conspire to really lead us into other downstream issues that are life-threatening, like cardiovascular disease, Alzheimer's disease, and so many more. We now recognize that while it's important to measure your blood pressure, look at your blood lipids, and certainly your waistline, that there's a very important biometric, a very important parameter that we really didn't know about, but yet is playing a central and pivotal role in these conditions, and that is uric acid. Now, many of you have probably heard of uric acid in the context of things like gout and kidney stones, but we now know that uric acid is certainly throwing a much wider net and is really very, very important. Now, the central figure in exploring uric acid and bringing this information to our awareness is Dr. Richard Johnson. Let me tell you a little bit about our guest today. Richard Johnson, MD, is the Thomas Burrell Professor of Medicine and the Chief of the Renal Division and Hypertension at the University of Colorado since 2008. He's a graduate of the University of Wisconsin in Madison with a major in anthropology and a graduate of the University of Minnesota Medical School in Minneapolis. He is a physician and a nephrologist, meaning a kidney specialist, whose research, which has been funded by the NIH, has focused on the role of sugar, especially fructose, in driving obesity, diabetes, high blood pressure, and kidney disease. And much of this work has explored the role of fructose metabolism, especially through its generation of what we're talking about today, and that is uric acid. In driving this phenotype, this dysfunctional metabolism, his work has included studies ranging from molecular biology to integrative physiology and even evolutionary biology, a fascinating topic that we're certainly going to talk about today. He's the author of some terrific books, including The Sugar Fix, which introduced the first low fructose diet, and also a book entitled The Fat Switch, which explores the role of fructose in driving the obesity epidemic. His new book that'll be published in 2022 is entitled Nature Wants Us to Be Fat: The Surprising Science Behind Why We Gain Weight and How We Can Prevent It and Reverse It. So really exciting to learn all of this about uric acid with our guest, Dr. Richard Johnson. Well, hello, Dr. Johnson. It's nice to connect with you today, and it's so great to have you here on the podcast.
Thank you, David. I'm delighted to be here.
I want to start by trying to determine how you got interested. The uric acid story is so exciting. It's so empowering, and I think that our viewers are really going to walk away from our time together today with a powerful new tool as it relates to some of these challenges that we have: weight gain, hypertension, dyslipidemia, type 2 diabetes, or at least insulin resistance. And I'd like to start off by asking, how did this all start with you? I guess it was 1999, wasn't it?
Yeah, it was. So I was very interested. I'm a kidney specialist, and so I was studying kidney disease. And one of the things about the kidneys is that they know that it has a very important role in blood pressure. And in fact, many people had pointed out that the kidney has a controlling role in blood pressure, probably by its effects on salt excretion. And so some studies had been done where they had taken people or animals with hypertension, and they found that if they transplanted the kidney from that animal to another animal, that you could transplant or you could transfer the susceptibility to hypertension. So, I was interested in why that was. And we were studying the fact, we realized that people with high blood pressure don't have normal kidneys. They actually have kidneys that have low-grade inflammation in it. And I had been thinking about it, and I realized that uric acid is a substance that it's the cause of gout, but there had been many, many papers that wrote that people with gout have low-grade inflammation in their kidneys. They have chronic kidney injury, very, very mild to moderate and sometimes severe. And so there was a study done in the 60s looking at patients with gout and looking at autopsies, and they found that kidney disease was present in something like 95% or higher. And so I started thinking about it and thought to myself, could uric acid be playing a role in causing subtle kidney damage that then might increase the risk for blood pressure? Now, at the time, uric acid was not really considered very important. It was thought to be a waste product, basically, and it was certainly important in the disease gout when it crystallized in joints, but it really wasn't thought to be important in kidney disease. And in fact, there were many papers saying that although it's associated with kidney disease, it probably doesn't have a causal role. And but we thought to ourselves, maybe maybe in patients who have gout or patients with high uric acid, small amounts of crystals could deposit in the kidney that could cause low-grade inflammation in the kidney, and that might make the kidney want to hold on to salt. So we started doing some experiments where we raised uric acid in animals, and so it turns out that humans have higher uric acid than other animals because we lack an enzyme called uricase, and uricase is an enzyme in the liver that degrades uric acid. So humans have a little bit higher uric acid than most other mammals. So in order to raise uric acid in a laboratory rat, we actually gave it this uricase inhibitor, thinking that we were going to get low-grade inflammation in the kidney and crystals, and that might cause high blood pressure. But what we found was that we did get high blood pressure. The animals became hypertensive when we raised uric acid, but when we looked in the kidneys, we didn't see any crystals at all. So
that must have been a huge aha moment for you and a challenge, because here you thought you were going to grow crystals in the kidney, it would irritate the kidney, and lo and behold, blood pressure would go up. But instead, that's not what you found.
Yeah, so I remember the day Marilda Mazzali was a Brazilian fellow who was in my lab, and she, and we raised the uric acid, so actually what happened first is we raised the uric acid, and she didn't measure blood pressure. She was actually looking at other things like kidney function and so forth. And we found that the kidney function really didn't change much, and the inflammation was pretty darn subtle. So we thought the study was negative, but because I was aware that in hypertension there was subtle inflammation, I asked her to measure blood pressure. So she went back, repeated the experiment. She was actually the only one in the lab who wanted to do it because everyone thought the study was going to be negative. And then she came back to me and says, Dr. Johnson, the blood pressure's high in these animals. I said, oh, did you do it right? Did you, what were the controls? And then we realized that we needed to do controls where we lowered the uric acid, like with drugs like allopurinol or other types of drugs. And so she did those studies, and then the animals didn't get hypertensive. And then we also did the thing where we raised the uric acid, and the blood pressure went up, and then we stopped the uricase inhibitor, so the uric acid levels came down, and then the blood pressure came down. And so we had to face the fact that uric acid might actually cause hypertension in the rat. And, at that point, it was known that 25% of people, 30% of people with high blood pressure have a high uric acid. And if you have bad hypertension, it's like 50 or even 75%. And so it was sort of, a remarkable observation because it suggested that we might have found a cause of hypertension.
Well, the story obviously gets a lot more exciting over the years when you block uric acid formation using a drug, allopurinol, in humans. For, I think, one study with adolescents who are overweight, and you have a powerful impact on blood pressure. So it looks like whatever the mechanism is, whether it's through nitric oxide, or we'll talk about the possibilities, But whatever that mechanism is in the rat is something that's actually happening in humans as well, and has given you a platform from which to argue that lowering uric acid is something to think about in individuals with hypertension. And as we continue our conversation, I think we're going to develop this theme across the entire metabolic syndrome participants. So you then did studies. I think one was published in JAMA on treating adolescents who were obese and pre-hypertensive. And how to tell us about that?
Yeah. So what happened was we submitted our paper saying that raising uric acid might actually raise blood pressure, and we had to be very careful. We sent it to Nature, and of course they were not interested because it was too provocative, really, because everybody had more or less thought of uric acid as being a waste product. And so we ended up publishing it in this journal called Hypertension, which is a nice journal. And then we figured out what did we need to do next. And of course, we actually validated our model and other animal models, but the key was to go to the patient, just as you say. So at that time, I moved in 2000. I moved to Baylor in Houston, Texas, and while I was down there. We started doing some studies, and we had another discovery, which was that what happens is when the uric acid, when you raise it, it causes inflammation in the kidney, and then that raises the blood pressure. And if you lower the uric acid, you lower the inflammation and you lower the blood pressure. But we realized that there was another thing going on, which is that if you kept the inflammation going a long time, then the kidney drove the blood pressure even when you corrected the uric acid. And so it was kind of there was actually this huge rain, and the medical center flooded the day after we finished our experiment. So we just barely made it because the poor vivarium flooded. But what happened
That's an
was
interesting aside. Who would have thought,
yeah, it was a
The whole future of uric acid
terrible
science
thing,
could have
but
floated away.
yeah, it could have floated away. But we finished the experiment like the night before, like at 8 at night. But and what we found, as I said, was that initially the high blood pressure was associated with high uric acid and was driven by the uric acid. But over time, the kidney took over, and then you could lower the uric acid, and the high blood pressure persisted. So what was interesting is that in humans, there's this fair amount of data supporting that uric acid tends to be high with new onset hypertension, and especially and then over time it can stay high or it can come down to normal. But basically we realized that if we're going to look to see if uric acid was important in hypertension, we would want to begin with studying people who have new onset hypertension, where it's not been so long that they start developing kidney damage. So around that time, I met this pediatric nephrology fellow named Dan Feig, and he had just become an assistant professor, and his function, one of his roles, was to run the pediatric hypertension clinic. And you probably know, many, many years ago, pediatric hypertension was mainly a congenital problem, kidney damage or
renal artery
if you're born with a single kidney or fibromuscular dysplasia. It was all what we call secondary hypertension. But as obesity started to increase, including in children, suddenly things like type 2 diabetes and high blood pressure started becoming more common in adolescents. So his clinic was filled with children, many of them who were overweight and hypertensive, who had true primary hypertension. So I gave him a very simple question. I said, why don't you just go and look and measure the uric acid and see what it is? And he came back, a month or two later, and he showed me this graph, and there was a direct relationship of uric acid with blood pressure in these adolescents. And what's more, it was only true in the primary hypertensive. So kids who had normal blood pressure had low uric acid, and if you had secondary hypertension, you may or may not have a high uric acid. But if you had primary hypertension, what we call essential hypertension, the hypertension associated with obesity, the uric acid was elevated. And it was like 90% of these kids had high uric acids versus like less than 5% of the controls. So Dan got this idea that he would actually do a randomized controlled trial to see if lowering uric acid could lower blood pressure. And he got an NIH grant with me. Actually, I was his mentor. It's called the K Award. And this launched his career, actually. And it was a double-blind, placebo-controlled crossover design where adolescents, I think their average age was like 14 or 15, who had just been diagnosed with high blood pressure, were randomized to allopurinol, and then there was a washout or placebo, and then there was a washout, and then they took the other drug or placebo. So everybody got treated both with placebo or allopurinol at different times. And what he found was it was unbelievable. These were kids who had never been treated with hypertension. They had never received any medicine at all. These were pharmacologically naive. They just had high blood pressure, recently diagnosed, and 90% of them responded. 88% responded to lowering of uric acid. If we lowered the uric acid under 5, they became normotensive. And in the placebo arm, only 5% or so responded. It was dramatic. It was like, oh my God, how could this be? Could uric acid be a cause of hypertension?
So again, it's an interventional trial in humans. You give a drug that inhibits an enzyme that's involved in the production of uric acid from precursors, and lo and behold, uric acid level goes down and blood pressure goes down. How was that greeted by the mainstream when you published?
I mean, it was viewed as quite exciting. In the pediatric literature, there was already some associations of uric acid with hypertension that had been observed. And of course, the association was well known in the adult, but I think there was a fair amount of pushback, but the study was so well done that it got into JAMA. And, so it had a high visibility. And it has subsequently been very, very highly cited. But at the time, of course, it hit with a splash, and not everyone probably viewed it as important as we thought it could be. But and then one of the other interesting things in that study, David, is we subsequently showed that one of the ways that lowering uric acid works is it works on a system called the renin-angiotensin system. And that's the same system that, ACE inhibitors are used for, angiotensin receptor blockers. So it's these drugs that block the renin-angiotensin system are used to treat high blood pressure frequently in many, many people. And lowering uric acid worked in part by reducing the renin-angiotensin system. And in this particular study, we measured plasma renin activity, which was high in these children, and it's known that new onset hypertension is often associated with high renin, and it fell quite significantly with treatment. So we had sort of a signal of the mechanism by which it was working as well.
Another mechanism that you've been describing for years has been the role of uric acid in terms of inhibiting nitric oxide functionality. And maybe we can segue first to a brief description of that role in hypertension and blood flow in general, because I know that's also something that's been studied. And that should also take us then to insulin resistance. So let's go through that.
Yes. Yeah, let's talk about that because that's exactly right. So we knew that the renin-angiotensin system was involved in uric acid, but we also had some experimental data suggesting that this system called nitric oxide was being inhibited. And as you know, nitric oxide is a vasodilator. It's produced by the endothelium. It keeps the blood vessels healthy. And the people who discovered nitric oxide, 3 of them got the Nobel Prize. So it's a very important system that keeps the body healthy. And it keeps the blood vessels dilated and blocks inflammation in the blood vessels. It blocks clotting. It's really an amazingly important system. And in young people, nitric oxide levels are high, endothelial function tends to be good. But as we develop kidney disease, or as we develop high blood pressure, or as we develop diabetes, this endothelial function is hurt. So what we started looking at, and we discovered that when the uric acid was high, endothelial function tended to be poor. And there was even a paper in the JAMA, I think it was in the JAMA, that looked at circadian rhythm, and they found that when uric acid goes up in early morning, nitric oxide levels fall, and there seems to be a circadian rhythm to it as well. And when we put uric acid on cells like endothelial cells, we could block nitric oxide. What was fascinating, David, is it does it multiple ways. It does it by working on the transport of arginine. It works on it by blocking the enzyme. It can scavenge nitric oxide itself. We found that it actually binds nitric oxide. I mean, it really has multiple ways of reducing nitric oxide. And our group wasn't the only group that looked at this. Other groups confirmed it, and there are multiple mechanisms. And if we can increase nitric oxide, it turns out we could actually, when we increase nitric oxide, we could block some of the effects of uric acid, such as, for example, hypertension. So we realized that uric acid was working on that, but inhibiting nitric oxide and activating the renin.
Over the past 100 years, fair to say uric acid levels generally have gone up quite significantly. And your work, I think, when you go online and read the description of Dr. Richard Johnson, it's all about fructose metabolism. And what then the segue into uric acid production. So let's talk about these parallels between rising uric acid levels over the past century, rising consumption of fructose, and I know that's going to take us to why we have this uricase mutation, or how it was originally beneficial for us. And ultimately, your book, Nature Wants Us to Be Fat, I think it'll be a great segue to that.
Okay, great. Yeah, so as we started looking at uric acid as a cause of high blood pressure, we realized that there are many, many studies that showed that a high uric acid independently predict hypertension, and it independently predict obesity and diabetes and all these things. And so it looked like it was really important. So I started thinking, could this epidemic of high blood pressure and obesity and diabetes be related to uric acid? So I became a historian, David. I spent my time going to the libraries and all. This was back in early 2000 when that's what you did. But I also, I discovered Google pretty early on as well, which is one of the most magnificent ways to look up old articles. And what I found was that this epidemic of obesity, although everyone had talked about it as starting in the 1970s, you could trace it back to 1890. That's right around 1890 was the turning point. I call it the tipping point. The obesity was seen in about 2 to 3% of people in 1890, and it was already rising by 1905, 1920, 1940. And the same thing was true for diabetes. Diabetes was like 2 cases per 100,000 people in 1890. That's what Sir William Osler wrote in his textbook, and then it started to increase dramatically into the 20s and the 40s and the 50s. And the same thing was true for high blood pressure. High blood pressure was actually rare back in 1905, and Janeway, who studied this, 500 people, said there was only like 3 to 5% of people had high blood pressure back then. And so there was this real dramatic rise over the last century. And I started looking at uric acid levels, and they also turned out, the trouble was there was different assays way back when, but around 1920 they started measuring it. And then a new assay came up in 1930, and that assay actually has been used routinely since then. And it's very easy to show that uric acid levels went from about 3 milligrams per cent in 1920 to an average of like 5.5 or 6 milligrams per cent like now. So it's been going up. So then the question is, why is the uric acid going up? Is it because it's going up because of obesity and diabetes and high blood pressure, or is it going up because of something else? And the big thing that controls uric acid, one of the major controllers, is diet. So I immediately thought, it's going to be purines. It's going to be. We've all been told that when I was in medical school, they said, well, uric acid is driven by purines, high purine diets. So things like beer, which has yeast in the beer, and red meats and things like this should be the primary driver. But when I looked, the red meat intake was actually flat, or actually tended to go down a little bit, depending upon which decade. I mean, it did not correlate at all. But I was aware, as you are, that sugar, yes, sugar, and particularly fructose, the component of table sugar, can raise uric acid. And, it was known by Osler. Osler knew that sugar raised uric acid. He said, avoid sweets if you have gout. This is like 1890. People were writing about it, but, it kind of fell off the map, and everybody was thinking it was purines when I was around doing this. And so the idea, the thought was that maybe could it be fructose? Could it be sugar? So I decided to do an experiment. And I gave sugar. Actually, it was done by Taka Nakagawa in our group. He was a Japanese visiting scientist. And he gave sugar to rats. And, over time, and he gave fructose to rats. And over time, they became obese and pre-diabetic, and they developed metabolic syndrome, but their blood pressure went up. And the original theory, David, was that we were going to lower the blood pressure, and that was the reason for this epidemic of high blood pressure. But what we found is that when we lowered the uric acid, the blood pressure improved. But guess what? The triglycerides were less in the blood. The insulin levels came down. The glucose levels were less. The fat in the liver was less. And even the weight gain was affected in the first study, not so much, but in subsequent studies, yes. And so we realized that when we were lowering uric acid, we weren't just improving blood pressure. We were like improving the metabolic syndrome. And what's more, as I looked at it, I became convinced that fructose and sugar was like the main driver of obesity. And this was like 2003. There weren't many people looking at it back then, to be honest. There was a group at UC Davis that had some grants and were studying this, but basically sugar had kind of escaped a lot of people's thinking in terms of being a cause of gout, for example, but also even as a cause of obesity. It was sort of suppressed at that point. And so we started doing studies to try to understand how fructose could cause metabolic syndrome and what was the role of uric acid. Because, everybody was saying that the reason you got fat was because you were eating too many calories and you're eating too much sugar. And uric acid is a metabolite, but it's like an offshoot metabolite. It's not like part of the fructose metabolism in the standard sense. It isn't like fructose is broken down to uric acid. What happens is that fructose consumes a little bit of ATP, and when it does, that degraded ATP turns into uric acid. So it's like a completely different pathway.
It's the rise in
So we said,
fructose
why would
consumption
that
that you assume then
different
led to the
pathway?
rise in uric acid levels with all of these detrimental downstream issues.
Yeah, exactly. So suddenly we started thinking, my God, fructose is actually playing a role in metabolic syndrome, and uric acid somehow driving part of it. So then the question was, how was the uric acid working? Because we didn't think it was just through the renin-angiotensin system, but we knew that it inhibited nitric oxide. And I was aware that mice that had endothelial nitric oxide knocked out developed features of metabolic syndrome. And so it's called the eNOS knockout. But basically, if a mouse doesn't make enough nitric oxide, they can develop features of metabolic syndrome. So we thought, wow, maybe that's how it's working. And as you probably, I'm sure, know, nitric oxide one of the ways that glucose is taken up in muscle, part of it requires a little bit of vasodilation, and the blood vessels have to be, dilated a little bit to help deliver the glucose to the muscle. And if you just inhibit that component, that can cause a little bit of insulin resistance. And so we actually did some studies like this, and so we found that insulin can stimulate nitric oxide in endothelial cells, but when we treated them with uric acid, it blocked the nitric oxide production. And so we began to realize that it could partly involve this pathway. But actually it turned out that it was a much bigger pathway than we had imagined. But when we first started, we thought it was just nitric oxide.
But going back to 2003, early in the day, that was a time when fructose was the safer sugar because fructose did not immediately increase insulin. Didn't require insulin for its metabolism. And it was, dare I say, even promoted by groups interested in giving information to diabetics. But the messaging was that fructose is the safer sugar.
Yeah, that's absolutely true. So when, table sugar consists of both glucose and fructose bound together. And the glucose, of course, stimulates insulin, and insulin can drive fat production. So there was a lot of thinking that the glucose component of sugar was the bad guy. And fructose actually doesn't stimulate insulin. And when you give fructose to an animal or to a human, glucose goes up a little bit, but it can often be delayed, and it's not very much. And so it's actually what we call a low glycemic carbohydrate. So the fact that it's low glycemic and it didn't stimulate insulin, it was sort of surprising that it might actually have a role in diabetes, because it seemed like it should be a safe sugar for diabetes. But when you give fructose to animals, over time they become severely insulin resistant. They become resistant to the effects of insulin, and the glucose levels go up. And so you end up with a high insulin, high glucose state when you chronically feed an animal fructose. But if you just give it a single dose, neither glucose goes up or insulin goes up. So it's a little bit of a paradox.
Yeah, this is really so very fundamental. And that is, while fructose doesn't acutely affect insulin levels measured in the short term, in the long run, you're setting yourself up for hyperinsulinemia, ultimately insulin resistance, paving the way for type 2 diabetes and all of the downstream relationships that it has. This is, man, if we ended our time together right now on that important point, I think it would be a job well done. But so critical to understand that this is a mechanism then that leads to elevated insulin and then relates to lipogenesis, the creation of body fat.
Yeah, we later were able to show that this path, we were able to dissociate the glucose from the fructose and causing insulin resistance. But I'm going to get ahead of myself if I go there right away, because
We got a whole bunch of bullet points that we've got to get to, but it's a question of getting them in order.
yeah, exactly. So, but yeah, so going back, so originally people were thinking that sugar was causing metabolic syndrome through the glucose insulin pathway. But it turns out that probably more important is the fructose pathway, because what fructose does is it makes an animal insulin resistant. And once you're insulin resistant, then glucose and insulin levels go up very high and will stay high even when you're fasting. So, so it probably is the fructose that's the bigger player.
Well, why on earth would it be a good thing for an animal to become insulin resistant? I teed that up for you perfectly.
Oh, yes, you did. You absolutely did. So it turns out, yeah, that one of the questions you're asking is why why do humans have a high uric acid? And why would we get a mutation that would raise our uric acid levels what I just told you is that a high uric acid can cause high blood pressure, it can cause insulin resistance, it can cause fatty liver, it can cause fat accumulation. This doesn't sound good. So why the heck? Why do we get this mutation that raised our uric acid levels? And that takes us back to when the mutation occurred. So again, there was a mutation in an enzyme called uricase. It happened around 15 million years ago, and that mutation occurred in our ancestors, and it was also the ancestors of the great apes and the ancestors of humans. There was a common ancestor at that point. And that animal had a mutation in uricase that raised its uric acid. And it happened around 15 million years ago. So I was very curious why that happened. And so I began by reading about what was going on back then. And one of the things that was apparent is that it wasn't just the ancestor to humans and great apes, but the ancestors to the lesser apes, like the gibbon, also had the same, or it was a different mutation, but it knocked out uricase around the same time. And there's a general rule: if you have a parallel mutations, there had to have been some kind of survival advantage that occurred that allowed these two mutations to take over these species, of which our ancestor was one of them. So it told me that there had to have been something that was a survival advantage back then to having this mutation, which didn't make a lot of sense because uric acid seems to be driving only bad things among us. So it turned out that around 20 million years ago was the beginning of the Miocene, which was this huge epoch. And the Miocene was this period of time where the first apes developed in Africa, or evolved in Africa. And the ape was this incredible breakthrough in primate biology because it had a big head and it was a bigger animal, and it was it was a smarter animal, and it lived in the trees back then, and it was tailless, didn't have a tail. These early ancestral apes were first in Africa, and they lived in rainforests, and in these tropical rainforests where they lived on fruit. That was their primary food. They ate almost exclusively fruit. It wasn't truly exclusively fruit, but 90% plus of their food was fruit. And during that time, they didn't have any predators. The climate was great, and for about 4 or 5 million years, they just did extremely well. It was like It was like Eden, you know. It was like this beautiful place where the apes were able to evolve into multiple species. And at one point, I think there were like over 10 species of apes. And then a bad thing happened. The climate began to change, and it started getting cooler. Instead of global warming, we had a period of global cooling, and the polar ice increased, and the sea levels receded, and it was a time when Africa, which had been separated from Europe and Asia, suddenly, as the water levels fell, a land bridge developed, and these apes were able to migrate out of Africa, and many of them went to Europe and to Asia. And at that time, the forest still had a lot of fruit, and they were able to continue to live kind of the way they normally did. But as it got cooler and cooler, the forest became more deciduous. There started being some open areas and savannas developing. And what happened was these primates had to come out of the trees to forage for food. There wasn't enough fruit. And a critical point in time was when the fig tree could no longer grow. And at that point, or this was in Europe, there wasn't able to, there was a loss of the fig. And so the fig was a very important fruit because it fruits all year round. And so when that tree died, there suddenly, during the cooler months, there was like not much fruit available. And the apes in Europe started to starve. Whereas the apes in Africa actually still did well. The forests receded, but they still had fruit all year round. So they kind of stayed the way they were. But for the apes in Europe, this kind of tremendous pressure to survive led to them to knuckle walk and to get stronger teeth and to start eating tubers and seeds and things that they didn't normally eat. And there was a massive period of evolution, and there were a lot of mutations. And it was at that time that the uricase mutation occurred. And we know, so there's a guy named Peter Andrews, who's this wonderful anthropologist, paleontologist who works out of the Museum of Natural History in London. And he's like the world expert on this period of time and the evolution of these apes. And I reached out to him and I said, I would love to meet with you and talk to you about, this hypothesis, why, about this mutation and what it might have meant back then. And he invited me, and I flew to London. I went to the museum and went back to this little room, and he had these old fossils of primates, and he would show them to me, and it was really an exciting day. But what came out of it was that these apes really were starving. But he had shown that it was one of these European apes that was our ancestor, and that by on the grounds of the structures of the fossils and so forth, the fossil evidence suggested that we actually evolved from a European ape, but that ape actually went back to Africa, right around 10 or 15 million years ago. And it went back to Africa because they were all starving in Europe. And eventually all the animals, the primates, died in Europe. And the apes became extinct in Europe, but one of those European apes went back to Africa, and we think it went with this mutation. So then we said, well, could that mutation have provided a survival advantage to these apes? It could have been one of the reasons it was able to survive in Europe as long as it did. And we realized that, if uric acid drives fat from fructose, that if the uric acid level would go up higher in response to fructose, that you might get a greater fat response. So one of my collaborators, Gabi Sanchez-Lozada, decided to do this experiment. So she took rats and she gave them this uricase inhibitor, which raised their blood pressure. Remember that? But then she gave that group a small dose of fructose, and she compared it to another group that just got the fructose without the uricase inhibitor. And lo and behold, the animals that got the uricase inhibitor plus fructose, even though they got the same amount of fructose, they showed a lot worse metabolic syndrome. They had more fat in their liver, they had higher blood pressures, they had worse insulin resistance, and it looked like the mutation was actually increasing the fat stores in response to sugar. And we said, maybe if you're starving and you're not getting enough fruit, what it's really doing, it's moving you from being a starving animal to having enough fat to survive. It's not making you obese, but it's preventing you from dying of starvation. And so we ended up working with a guy named Uric. And Uric is this brilliant molecular biologist who can actually resurrect extinct genes. And he went and he resurrected the extinct uricase from these early apes. And then we put that uricase into liver cells, human liver cells that don't have uricase. And then when we exposed them to sugar or exposed them to fructose, what happened was in the normal cell it made a little bit of fat, but the uricase was there and the uric acid levels weren't very high. But when we put fructose on liver cells in which the uricase enzyme had been knocked out, we got this whopping response with sugar. Of fat. And so basically, what we were able to show is that when we had that mutation, that it allowed animals to put on fat easier in response to the same dose of sugar. So for the same dose of sugar, you get a bigger fat response. You get a bigger insulin resistant response. And this happened to us, humans, 15 million years ago. So a mouse can eat a fair amount of sugar before it gets fat. And you can make the mouse fat, for sure. But the relative same amount in a human would make it extremely fat because we are sensitive to sugar.
And we're sensitive by virtue of carrying this uricase mutation. And I just hope our viewers are getting that here is a guy who found some things in laboratory rats, was thinking about it, and the story goes back 15 million years ago in
Yeah,
terms of an evolutionary advantage at that time during caloric scarcity that allowed survival of the fattest, though they weren't actually probably not fat, but that we're the legacy partners in this agreement, that we carry this genetic situation whereby with exposure to fructose, we increase uric acid production. It's a danger signal that we're about to reach a time of caloric scarcity. Better make the fat, better become insulin resistant. When you say nature wants us to be fat, the title of your new book, you really mean our genetics, our nature. Our genetics
yes,
is programmed to keep us fat for the winter that never comes, the caloric scarcity that we never encounter.
exactly. Yeah, and it turns out that when you look at the features of metabolic syndrome, as you were brought up earlier, actually, fat, of course, is a form of energy. It's a calorie. So if you're starving, we live off our fat. So if there's no animals will store fat so they can survive the winter. A hibernating animal or a long distance migrating bird will increase its fat so that it can survive when it migrates. But they don't just increase their fat, they also become insulin resistant. The hibernating bear and the long distance migrating bird, they become insulin resistant as well. And the reason that is not because they want to become diabetic. They don't want to become diabetic. But when they become insulin resistant, they're preserving glucose for the brain. So what happens is uh, the muscle likes insulin, needs insulin to move the glucose into the muscle. And the liver does too to some extent, but particularly the muscle likes, needs insulin to drive glucose into the muscle. The muscle can use a lot of our glucose, but the brain can use a lot of glucose too. But most of the brain does not require insulin to get into the brain. Most of it. There's areas of the brain that do require insulin, but most of the brain does not. And so what happens is if you become insulin resistant and you don't have a lot of glucose around, you're actually allowing that glucose that you do have to be a preferred fuel for the brain, because the muscle is going to kind of be resistant to taking it up, so there's more of it around to go to the brain. So insulin resistance is it's a tactic an animal does to try to help it,
It, it,
maintain fuel for the brain when there's not enough food around. So one of the things that happened, David, was when we realized that the mutation was probably a survival move to help these animals survive starvation. Then the question was, well, how high does the mutation itself take the uric acid? And could it be that the mutation takes it up a certain level, and then all the sugar and everything we're eating takes it to the levels that we are at today? So I thought to myself, what we really need to do is to measure the uric acid levels in like the great apes. And, are their uric acids 6 or 7 like us, or are their uric acids lower? And what we found was that I went to the San Diego Zoo and I met with Bruce Rideout, who's a guy that he's a great scientist who works there. And we ended up doing a study where we measured uric acid, all these giant, gorillas and all this stuff. And the blood was sent to my lab, And I'm supposed to be a kidney doctor, but I got these bloods from gorillas, and so it was quite entertaining. But basically, their uric acid levels were like 3. They weren't 7 or 8. They were like 3 or 4 milligrams percent. And the monkeys and stuff that had the uricase, they had uric acid levels of 1 or 2. So the mutation only took the uric acid from 1 or 2 to like 3 or 4. That's what the mutation did. And we wanted to verify that. So I wanted to find a group of, some people who are not on a Western diet, people who are on some kind of native diet. And I was reading about these Indians called the Yanomami Indians, which are this native Indian group that lives in southern Venezuela in the jungle, and they eat a lot of plantains and tubers. Plantains have fructose, but they eat a lot of vegetables and a little bit of meat, but they're not eating refined sugar or things like that. And so I thought, I gotta find a way to get some blood on these people. And I saw that Jim Neal, the guy and others who had written about this kind of idea of a thrifty gene and of a time, a time in the past when we were starving and now we're getting fat from it. And he had done a study of the Yanomami Indians with a guy named Bill Oliver, and they had published a paper where they had blood samples on these Yanomami Indians. So I contacted. Neil had passed away, but Bill Oliver was still alive, and he was he had been the head of pediatrics at University of Michigan. And I reached him, and he actually had these samples of Yanomami blood in his garage, in a freezer in his garage. I go, you're kidding? And he had IRB approval to share the samples with me. So again, we got these blood samples sent to us from Yanomami Indians collected like 20 years before, and their uric acid levels were like 3, just like the gorilla. Whereas the members of the party that had gone down there, like Jim Neal and Bill Oliver's blood, their uric acids were like 5.5, 6. So they were kind of like a normal Western diet, but the Yanomami Indians had a uric acid of 3. So then I was able to say, aha, I'm going back now to this epidemic of obesity. And around 1920, humans had people living in America and England had uric acid levels of 3 or 4. And now everybody's got higher levels, and we've changed every few years. There's a change of what's normal because as the uric acids go up, people just adjust what's normal. But the true normal is probably around 3 to 5 is where it should be.
Interesting that in addition to James Neal's work in 1961 was the book by Alexander Haig in 1896, focusing on the central or important role of uric acid as a causative player in so many diseases well beyond gout and kidney stones.
Yeah, I got to show you this one thing. 1 second. I don't know if you can see this, but this is a when Haig published his book, for about 15, 20 years, people were thinking that uric acid might be the cause of obesity, high blood pressure, kidney disease. And this is an old ad from France, from Paris. And basically, it's a guy who's trying to clean the kidneys of blood pressure, of clean the kidneys and make the kidney healthy. And he says here how he's going to treat arterial sclerosis, obesity, migraines, gout, and everything by lowering the uric acid. The only problem back then was they didn't have any treatment that worked. So, but they were thinking about it.
And then it got lost. It was, I guess, too much to get your arms around. But, it's just our nature to want to pigeonhole things like uric acid into one area, end of story. Like NAD. We all learned about NAD and thought, well, that's really interesting in mitochondrial energetics. End of story. Now we look at the role of NAD in relation to methylation patterns, et cetera, and DNA repair. That said, though, I want to just explore for just a moment then that this represents, vis-a-vis James Neal's work, the thrifty gene, that this is a classic environmental evolutionary mismatch whereby we were gifted this powerful gene, a change in gene, i.e. change in your case functionality as a survival mechanism that's now responding in a phenotype that's really quite threatening to us in terms of gaining weight, becoming hypertensive, and all the other aspects of metabolic syndrome that you've described.
Right. So I do think that this is one of the key problems.
In that the great apes who consumed a lot of fruit nonetheless and have the uricase mutation nonetheless have as you've demonstrated, uric acid levels that are significantly lower than humans, it means then maybe there's something not quite as threatening from a fructose perspective with regards to eating fruit in comparison to fructose sweetened beverages, fruit juice, for example.
Yeah, so we've actually done a number of studies that have looked at this. So one thing we realize is that fruit, a whole fruit has maybe 4 to 8 grams of fructose. Well, when you drink a soft drink, you get 3 times that. So and when you drink a soft drink, you drink it really fast. People tend to drink it fast, so the concentration is based upon not only how much you eat, but how fast you drink it. And so the concentrations of fructose are very high when they hit the liver, and that triggers this big reaction that with the uric acid, and that drives all this pathway. But when you eat a whole fruit, you've got the fiber, you've got less sugar, it takes longer to eat, it takes longer to absorb it, and it also contains all these good things like flavonoids and vitamin C and things like that can block some of the fructose effects. And so what happens is you don't get as much of the sugar, but you do get a lot of these good things like vitamin C and epicatechin and other interesting flavonoids that actually can counter some of the fructose effects. So
And some of these flavonoids act in a similar mechanism as does the actual pharmaceutical allopurinol by inhibiting xanthine oxidase. Things like quercetin, luteolin can actually help us reduce our production of uric acid.
yes, that's right. Also, they block some
Now,
of the fructose metabolism themselves. So we worked with luteolin and quercetin and fisetin, angosteen, osthol. Yeah, so there's a lot of these components that are in fruits that are good. And here's a very interesting thing. So animals, when they want to get
fermented.
fat, they'll eat fruit, but they'll wait till it ripens even more. They like it when it's almost a little mushy. And what happens is when that happens, the sugar content goes up and the vitamin C content goes down. So it's like the plant wants you to wait. They want the animals to wait till the seeds are maturing so that they can that when they eat the fruit, they'll disperse the seeds and the plant will grow. So what happens is the animals will wait till the fruit's kind of riper, and we tend to like tartar fruit. And the other thing is like animals when they'll in the fall they'll eat huge numbers of fruit at one time. So like a bear can eat 10,000 buffalo grapes in a 24-hour period. So they eat huge amounts, and so they get a load of fructose, and it's low in vitamin C, so they can engage this pathway to help them put on fat. But for humans, when we eat fruit, we're eating usually maybe a couple of fruit perhaps at a time, but often we don't eat a lot of fruit at one event, and we're eating that with all these good things. So we did a study in Mexico where we put people on a diet low in added sugars. It was low in refined sugar, it was low in high fructose corn syrup. And one group we gave, and so they were on a low fructose diet. They were also with mild caloric restriction, which I wish they hadn't done, but they did that. And the other group had the mild caloric restriction and also the low refined sugar, low high fructose corn syrup, but they were given fruit supplements. And what we found is both worked to reduce the metabolic syndrome. And in fact, the one with the fruit supplements tended to do a little bit better, just a little bit better. But basically, the including natural fruits did not block the ability of the to improve the metabolic syndrome. Having said that, I have personally had people call me who are drinking fruit smoothies or fruit
I hope that's
juices,
not real
or
bad.
just
I'm
eating
guilty
huge
of that
amounts
in the
of fruit,
past.
like a bowl of grapes at one big sitting. And yeah, well, I think we all do it. But if you eat a lot of fruit at one sitting, you can overcome this the benefit of the fruit. And fruit juice, where you can drink like 20 grams of fructose rapidly, and you've removed a lot of the fiber and stuff, that may not be so good. So
And
the
diabetes.
epidemiologic studies suggest that fruit juice can increase the risk for obesity, whereas natural fruits don't. So bottom line is 90% of the time, if you eat natural fruits, you're going to be okay. But if you drink fruit juices and so forth, or if you eat a lot of high fructose fruits at one sitting, it's probably not so good.
But I think that the take home point here is that the three inputs that we should be concerned with are purines from certain types of small fish, scallops, organ meats,
Yep,
organ meats,
right.
fructose, of course, which is ubiquitous, with close to 70% of the foods, processed foods on the grocery store shelves in America now with added sweetener, much of that derived from high fructose corn syrup. And the third player would be alcohol, also enters into the similar metabolic pathway, utilizing ATP to AMP and creating uric acid.
Yeah, absolutely. So beer in particular is the of all the types of alcohol, it raises the uric acid the most, and it's associated with the development of metabolic syndrome. There's many studies that link beer drinking. You know, it's sort of thought that the beer belly. So I mean, in the lay population, or the lay public thinks of beer as being a cause of obesity, but in the medical literature, it's sort of not been talked about that much. But the studies that have come up validate what people think, and that is that beer tends to be the most fattening of the different types of alcohol.
And yet it's marketed as being as light beer because it has low calories, and yet it's the purines and the alcohol.
Right, it's the brewer's yeast. In fact, non-alcoholic beer also increases the risk for raising uric acid and
There are now purine free beers available. Japan has really taken a lead on that, and you can get beer that's purine free and beer that's low alcohol. I don't know if that you can get that in the same beer, but I guess mix and match.
yeah, no, I know they have that low purine beer in Japan. I've been told by some of my Japanese friends that it's not that tasty. Have you ever tried it?
And if you have to drink beer. But the point I'm making is that it seems quite clear, as I'm certain your new book is going to talk about, that we can engage dietary and other lifestyle changes to have a pretty significant effect in terms of lowering uric acid, and therefore this pivotal mechanism that you have now described as playing such a central role in so many of the issues we're trying to avoid.
Yes. So, and also, David, one of the interesting things about these foods is that uric acid is not static. If you drink beer, for example, your uric acid levels will go up within 2 hours. If you drink sugar, your uric acid levels go up in 15, 30 minutes. So it's the what we call postprandial, or the blood levels after we eat can go up significantly. So the idea that, when people would say, that they had a dinner of duck, beer, and, some sweets for dessert, and then they woke up in the middle of the night with a gout attack, it may actually be related to the fact that the uric acid rises after they eat a meal. And so if you look at the area under the curve over a 24-hour period, that how high the uric acid is isn't always reflected by the serum uric acid in a fasting state. And so even if your fasting serum uric acid only goes down like from 5.5 to 5.2 by restricting purines and sugary foods and so forth, and alcohol, you may be blocking this great sweep, this great rise of uric acid that can occur after meals, and so there may be a lot more benefit than you would expect just for the small change in serum uric acid in the fasting state.
Mm. So again, it's kind of an area under the curve discussion that I think our viewers are probably well aware of, who are understanding why continuous glucose monitoring, for example, is valuable in terms of looking at insulin, glucose, metabolism, et cetera, that it's the time that something is, how high it's elevated, and times the time that it's elevated. How long does it remain elevated? So the number we should be shooting for in terms of keeping our uric acid below would be?
Well, so in terms of the relationship of uric acid and blood pressure, it's linear for a long ways. But when it comes to things like diabetes and obesity and risk for kidney disease, it does seem to be that around 5.2 to 5.5 is like an inflection point. So, personally, I would like to keep my uric acid at 4, but if we can get it to 5.5 or 5.2, depending on which study, that seems to be where there's an inflection point. And so once you're in the 6s and 7s, the risk for developing disease like hypertension and diabetes starts to go up pretty significantly. And then when you get around 8, it goes up even more. And then when you get up to 10, it's like going up, asymptotically. So I would say, I'd be interested in your thoughts too, David. What do you think? What from you're an expert, you become an expert on this.
Well, I think that there's going to be I think there's going to be some implication with respect to things like microbiome effects and microbiome input into the downstream effects of uric acid. I think we're going to learn what SNPs may play a role in terms of how we respond to various levels of uric acid. My mission in life has always been to kind of give the overriding kind of large net general ideas. And I'm saying that men, women, and children 5.5 and below is reasonable. Is it ideal? Probably not. Is a fasting blood sugar of 95 good enough? Probably not. We can go lower. Fasting insulin level of 8 is what I recommended years ago. Could be lower. But I think I want to get as many people, involved in understanding the role of uric acid now as a very important biomarker. And I think giving a reasonable goal, it will help people achieve it. And so when you and I first started talking, I told you my level was 5.5. It's now 4.6, which I think is a significant drop. And it's just, in my case, the recognition of the the anchovies and the sardines, which were a daily event. I don't drink much alcohol, and I have whatever fructose I get is via eating fruit. But I do want to leave our viewers the clarity on the fruit that contains fruit sugar, fructose, that an apple a day is not going to bring the doctor around, right? An apple a day or two, perhaps. Apples these days are bred to have more sugar, to be sweeter, and to be larger. So, maybe it's one apple a day.
Right.
But I think that we have a good ability these days to home test our uric acid levels. And I will tell you that, as you probably know, that we may see in the future the ability to continuously monitor our blood uric acid levels as we do our blood sugar, which I think is going to be a phenomenal addition to how we understand our personal physiology, our own precision medicine. So I think, we're just at the beginning of this understanding of the potency of this biomarker. And again, it gets down to how you respond. How do I respond to a given meal? I think we're going to see that fructokinase activity, et cetera, is going to vary based on genetic polymorphisms, and therefore people are going to tolerate things to a different degree. But I think by and large, the general ideas are, your uric acid is elevated. You got to watch the fructose. You certainly got to watch the alcohol and the purines. You want to add in some quercetin. You want to consider luteolin, vitamin C, tart cherry. Make sure you're hydrated. Make sure when you check your uric acid level that you haven't come off a 3-day fast, or you just haven't done some overly intense exercise. All the things people should be thinking about, checking at the same time. And if you're using a home uric acid monitor, I think that should be standardized against a good blood test that you might get at your doctor's office. So I think there's a lot here. And I think it's just another example of how we pigeonhole something like uric acid. I received the same training you did. It's gout and it's kidney stones. And it's gout caused by, these fat men drinking beer and eating, these very purine-rich foods. And it's almost historical, save for the very small percentage of people suffering from gout today whose uric acids are very high. And now to have this powerful tool that you, Richard Johnson, have put in our toolboxes to help those people who can't lose weight, who can't bring their blood sugars under control, regain insulin sensitivity. I think it's a new dawn, and you've done some amazing, amazing work. And as I told you before, it's the reason I reached out to you. I learned about your work in a Peter Attia podcast when I was on a run one day, and it's been life-changing for me, so I appreciate that.
Well, thank you very much, David. Your words are very wise about the goal of trying to get the uric acid down, and that it may require a personalized approach. And I agree with
Great.
you. 4.6
Well,
or 4 to 5 would be ideal, but I also agree that something like 5.5 is a really big target. So, yeah, I think that you're right. I mean, we did this one. Everybody says that uric acid may be high because you're obese or because you're diabetic, and so there's a lot of people who think it's the direction of causality is the other way. But I will tell you, we did do this one study where we took people. This was a study in Japan, but we took all these people who were lean, did not have diabetes, did not have high blood pressure, did not have high triglycerides. Basically, they were healthy, and the only thing they had was a high uric acid. And that group, we didn't have to worry about confounding factors. None of them were overweight. And that group showed that they had an increased risk for developing obesity, diabetes, high blood pressure, chronic kidney disease in the next few years compared to people who did not have a high uric acid. So,
but to be fair, there is a feed-forward cycle as it relates to insulin in the kidney with respect to uric acid.
yes, that's true too.
So this does make the matter worse with time.
Yes, that's absolutely true.
Well, thank you for spending time with us today. And what a story about a guy who gets interested in something, then studies, our primate ancestors 15 million years ago, writes an amazing article in Scientific American, or was written about your work. And I would encourage all of our viewers to take a look at that. But again, we will connect as we get closer to your book launch. And I'm really excited about it. So again, thanks for being with us today. Well, this is exciting. This is a very important point for us all to understand this pivotal role that uric acid is playing as a driver of obesity, as a driver of hypertension, a critical player as it relates to insulin resistance and type 2 diabetes, and as we talked about, even brain decline in terms of shrinkage of the brain and declining brain function. So really looking forward to Dr. Johnson's new book. It's an exciting time ahead. I'll be back soon. I'm Dr. David Perlmutter. Bye for now.